Hello, I'm Koki Mimura from QST in Japan. I'm far away, but very happy to be able to attend in this way. I would like to thank all organizers for this opportunity to present my work. Today, I'll talk about current status of our ongoing study about common marmoset behavior analysis entitled Unsupervised Temporal Segmentation of Marmoset Goal-Directed Behavior. So our research question is, how can we understand social communication? What is really happening in communication? How can we evaluate the dynamics of contextual interaction by multiple agents? And what kind of framework does the social brain plays for such real-world social behavior? These issues are still very challenging. In recent years, research on human communication has focused on verbal aspects such as natural language processing. But as people say, eyes are the window to the soul, and like that, nonverbal process is also important. And we know this, which is why we conduct our online meeting via video chat instead of phone call. In natural communication, we encode our internal states as motion and posture and decode them in the other person. How can we approach such nonverbal elements of social communication? One approach is quantify and computationally analyze the movement information. With the development of artificial intelligence technology, the technical hurdle for tracking human and animal motion has been lowered. This slide shows a typical motion tracking algorithm that has been reported in recent years. And however, it is still difficult to extract the internal state of agents involved in communication from the tracking results. In other words, there is a big challenge of how to statistically modeling this behavior information in a data-driven manner. This is one hypothesis to tackle this issue. If our behavior has a contextual structure that encodes our emotion and intentions, it could be analyzed by an algorithm similar to the syntactic structure of language. This is a powerful idea if we assume that language is a data-derived communication method and its origin is in nonverbal communication. If so, can we statistically estimate the syntactic structure into the behavior, like language processing, and decode agent internal state during social interaction behavior? This is also important in terms of dealing with social dysfunction. For example, autism can be diagnosed at 2 to 3 years of age. In this case, the subject is assessed for the risk of social function based on a behavior histogram as shown in the figure, and just based on the professional observation. Of course, this kind of professional observation is a very important method, but as you know, there is room for improvement in terms of objectivity and reproducibility of screening. Understanding of social function that focused on nonverbal elements will make an important contribution to these clinical studies. This is also an important issue to answer the question of what social functions are. Okay, now the direction in which we need to work has become clear. Let us consider describing goal-direct motion in a state transition model with a hierarchical syntax structure such as segment or chunk. And the social communication is described as a model that the interaction effect between agents as described in this figure. And in this model, social dysfunction is addressed as a type of syntactic disturbance. There may be change in the frequency of occurrence of segments, or there's a specific chunk may be observed, and or there may be irregularity in encoding and decoding of interaction or transmission. If such disturbance could be estimated computationally, then linking them to abnormality in social brain function would reveal many aspects of social function. This is a long-term strategy of our research, and in short term, We are proof of concept experiment this model in non-human primates. This is called marmoset, a small New World non-human primate, and they are known for the comparable brain structure and function and high functional social communication skill. For example, they have a mirror neuron system and the patch area specifically excited peer face images on their visual cortex. And on the behavioral aspect, they are known for understanding third-party reciprocity and learning from the imitation of others and sharing food with family and peers. This is a demonstration of marmoset food sharing. There are two adult marmoset and they are not family, but share the two pieces of food one by one without fighting. The process clearly involves eye contact and synchronized behavior, indicating that the common marmoset communicates its intention to others through their motion information. By measuring this simple goal-directed behavior in detail, we can try to statistically estimate the temporal syntactic structure of their behavior. Returning to the strategy overview, the advantage of using a model animal is that we can use cutting-edge technology to manipulate neural activity. This will allow us to clarify cause and effect relationship and map social brain function to contextual disturbance in social behavior. We have succeeded employed chemogenetics to transiently and minimally invasive manipulate neural activity in marmosets. This video is a demonstration of chemogenetic neural manipulation. I'm not going to go into detail here, but you can find a preprint on bioRxiv. You can access it from the QR code. So if you have interesting, please access it. So our research strategy have 4 steps, and the first step, neural manipulation, we have already succeeded in developing neural, chemogenetics. So today, we will talk about the following 2 steps. First, the development of an original marmoset motion tracking system, and the second, quantitative evaluation by unsupervised machine learning on marmoset behavior sequence. This figure shows a schematic overview of our original motion tracking system. The animal's behavior was measured by 4 depth cameras, and a skeletal model was fitted to the 3-dimensional coordinate. Furthermore, by training the object recognition algorithm called YOLO, or marmoset faces, we succeeded in estimating the 3D information of face orientation without body markers. This is a demonstration of the final version, and in the left panel, the estimated parameter overlaid on an image from one of 4 cameras, and the right panel shows the horizontal and vertical coordinate respectively. You can see the position of the body parts and the orientation of the face are well estimated. Main part of our system was developed by our collaborator, Dr. Matsumoto, and the system is freely available from the URL. And so let's move on to the next topic, segmentation algorithm of marmoset behavior sequence. Now we have detailed marmoset behavior parameters, and we would like to somehow describe the data as a discrete level assigned status transition model. Therefore, we employ the Gaussian process and Hidden-Markov model, an unsupervised machine learning algorithm. The Gaussian process is responsible for nonlinear regression and clustering the trend. By using this algorithm, the principal component score data for the behavioral parameters shown in the right panel, we was split and clustered into 11 labeled status shown in the left panel. In other words, a marmoset whole body motion recorded over 1,000 seconds were described by an array of 11 different segments. Let's take a look at the detail of the data set. And one data set is 20 seconds of motion tracking data, including 1 or 2 feeding. We recorded totally 51 data, so there is 1,020-second records were used for this analysis. Feeding involves 3 patterns, and they are taking food set on the wall by hand, taking food set on the floor by putting face close to it, and reaching out to take food set on the floor. The behavioral parameters were rescaled from the center coordinate to the body origin, and the principal component analysis was employed for dimensional compression. There is a graphical model of our Gaussian process and hidden semi-Markov model. The bottom row is a series of principal component scores, and the top row represents the discretized status, and with a Gaussian process in between. Here is a demonstration. On the left is the result of motion tracking, and on the right, the top is scaled behavior parameters, and the middle is the principal component scores, and the bottom shows the transition of segmentations mapped by color and y coordinates. The timing of feeding is indicated by blue vertical line, and as you can see, in this case, the segment number 3 represented in orange horizontal bar contain the feeding timing. In this figure, each segment is shown divided into clusters, and you can see the sequence with the same trend are classified as the same segment. And then we counted which cluster contained the feeding timing. And the trend was clear that each feeding subtype attracted into different clusters. These are some example. As you can see in this video, number 3 segment indicated feeding behavior with their hand, and number 10 segment clustered by approaching by their face. And number 5 segment represent as well as wall feed context. Actually, as you can see in the videos, there is clearly a correlation between the segment, which means before the number 3 feeding segment, there is a number 10 segment to approaching to the food. And after number 3 segment, there is a number 6 segment indicating food handling and eating situation. So could we quantify the contextual chunk structure on the marmoset feeding behavior? To this, we check the transition probability. If there is a correlation between pre-post segment transition, there could be some cluster transition relationship. Left panel described all segment transition map, and the right show feeding related context. The color indicated subtype of feeding behavior, and as you can see, those 3 subtypes represented clearly different cluster of segment transition. This is network graph representation of segment transition. A large number segment including feeding timing, and the arrow indicate transition probabilities. It is clear that these 3 types of feeding has an independent chunk structure, which means subset of segment sequence that appear in a specific type of behavior. Okay, today I introduce our current status about marmoset motion segmentation analysis, and we examined pattern status transition in goal direct behavior of marmoset, and showed data-driven chunk structure according to feeding subtypes. We already cleared the first 3 steps, and now checking how it works on social feeding situation in manipulation of amygdala neural activity. So we hope we can tell you the result of the next time, hopefully in Paris. And thanks all our collaborators and funding supports. And thank you for your attention. Please give us a comment or question. So thanks again.